the diffusion osmosis lab. Okay, and so in this lab what we would be using is what's called a gelatin tube. So gelatin is like what's found in a Jello, right? You put the gelatin in this tube. It acts as a medium, and we're using it to see how diffusion works, right? So the concept of diffusion, I like to use the perfume bottle as a good example, right? If you took some perfume, it's a liquid, it vaporizes, and that's what you smell, right? So perfume can leave the perfume bottle if you open it up in a room, and it would diffuse into all the air in the room, and it would go from the high concentration in the perfume bottle to lower concentration into the room. And it would continue to diffuse in the room until all the perfume left the bottle and all the air in this room, right, had an equal amount of perfume, right? At that point, it's no longer, there's no concentration difference, right? So diffusion's only going to occur when there's a concentration change, and it'll go from high to low concentration. It only requires time. That, you know, it requires a few other things, but the main thing to remember, right, it just takes time for that to occur. You don't need a membrane, for instance, for diffusion. It can occur in the atmosphere, right? So if we're looking at osmosis, it's diffusion of water specifically across the selectively permeable membrane, but a membrane would be required for diffusion. It would require time, for osmosis, it would require time as well as a membrane, right? So that's a little bit diffusion. It can't occur across the membrane. It's going to occur inside the gelatin on these tubes. So what we do for this experiment is we want to vary a couple things. We want to use different, say, dyes that have different molecular weights. So I think one has a molecular weight of about 300, one has a molecular weight of 500, and one has a molecular weight of 700. You know, that's basically a way of telling you how big the molecule is, right? The lower the molecular weight, the smaller it is. So if it's having to diffuse through this gelatin, which is a protein, but it kind of gets in the way, it will slowly diffuse down the tube. I'm sure gravity helps it to some extent. But you would expect the smaller the molecule, the smaller the molecular weight, the easier it would be to diffuse, right? The heavier it is, it just takes more work, right, because it's heavier. So you'd expect as molecular weight increases, it would slow down diffusion, right? So the dye, it starts at the top of the tube, and then it would diffuse down the tube over time. So the yellow might come down to here. Then we would measure that distance, after an hour and a half, and you'd expect the smaller, lower molecular weight to go down the tube more quickly than the heavier ones. And that's usually what happens. Then the other thing we do is we keep 3 tubes with 3 different molecular weight dyes at room temp, and then we put 3 tubes keep it on ice so it's colder to see what effect temperature has. So you would expect things to be able to move from a high concentration to a lower concentration more quickly as the temperature increases, right? Because basically the temperature is telling you how fast molecules are moving. So if the temperature in this room was 20 degrees Fahrenheit, it's below freezing in here, then the air molecules would be moving more slowly than they would be when we've got the temperature where we normally have it. But that's what the temperature tells you. It gives you a relative idea how fast the air molecules are moving. Okay, so if we put these 3 tubes on ice and then waited an hour to see how far the dye went down, we'd expect to see the movement to be less than it was if we kept them at room temperature, at a warmer temperature. A warmer temperature, they would move more quickly. Okay, so these gelatin tubes are used to, as a way, you know, a model for diffusion inside gelatin. You know, it's not the optimal, you know, say experiment, but, you know, gelatin's cheap, it's easy to make the tubes. And then we can ask, you know, questions about what molecular weight has, what effect molecular weight has on diffusion versus, and then also see how temperature, the higher the temperature, the more quickly you'd expect diffusion to occur. Gelatin tube diffusion. For osmosis, we're using what's referred to as dialysis bag or dialysis tubing. Okay, so if you saw this picture on the practical, right, this was used, apparatus was used to demonstrate osmosis using a dialysis membrane. So the membrane over here, you show, I use a little square to represent that membrane. It's like a hollow tube. We tie off the ends, right, and then we fill it with like 10 mils of water, and then we can, and some of the bags would contain sucrose as well as water. Now the bag itself is semi-permeable, right? It means some things can get in, some things won't get in and out. So you make the holes in the bag. I draw little circles here to show them. They allow water small enough, right, to, let's say it's a little circle, it can get in and out of the bag. Water will move in and out, or, you know, use osmosis to get in and out of the bag. If we put sucrose in the water as well, the sucrose is too big to move through the dialysis membrane or bag. Right? That's why it's considered semi-permeable. So if we put this bag into a big beaker, and in the beaker we just had water, and then we had water inside the bag, the water in the beaker could get into the bag, the water in the bag could come out of the bag and go into the beaker. Right, can go back and forth, right? We just put this bag containing sucrose into this water into this only water beaker. So what happens over time, you leave that in there for an hour. We would have weighed the bag before we started the experiment, and it usually starts out and weighs about 10 grams because we have 10 mils of water in it and a little bit of sucrose. So what happens over time is water goes from the beaker and into the bag, and then there's water in the bag that goes out of the bag into the beaker, right? That's the osmosis. The problem, because the sucrose can't get in and out, it stays in the bag, but it ends up blocking some of these holes. So water can't get out when it does, but water can still get in. Okay, so there's a whole lot of sucrose in here, right? That's what happened. So over time, more water can osmose into the bag than can osmose out of the bag, because the ones trying to get out of the bag are blocked by sucrose. Okay, so it goes from being 10 grams at the beginning, and sometimes it can weigh as much as 15 grams at the end, because that means 5 grams of water got into the bag, and the bag weighs, it's heavier. Okay, so the dialysis bag or tubing is semi-permeable. The sugar molecules or sucrose is too big, that's why it can't get out of the bag. The water molecules can get in and out, no problem. And we refer to this is used to demonstrate how osmosis can, right, diffuse, water can diffuse in and out of or through a semi-permeable membrane. Okay, so that's it for the osmosis diffusion lab, and